Good morning and welcome to the Florida Health Grand Rounds. We are excited today to welcome Dr. Giuliano. A few things that you should know prior to starting the seminar today. All phone lines are currently muted, and please enter any questions that you may have into the question box on the control panel. We will have time for questions and answers at the end of the presentation. For your convenience, the slides for today's presentation are in the handouts tab if you have any problems with viewing. And for those of you who would like to receive nursing continuation credits, please fill out the survey that will pop up at the end of the webinar and be sure to enter your correct license number. With that, we're going to go ahead and get started with introductions for the seminar today. Dr. Giuliano is the founding director of the Center for Immunization and Infection Research in Cancer at Moffitt Cancer Center in Tampa, Florida. She received her doctorate from Tufts University, Boston, Massachusetts, continuing her studies at the New England Epidemiology Institute in Boston and at the University of Arizona in Tucson as a National Cancer Center Prevention and Etiology Fellow. From 1993 to 1998, she was the recipient of an NIH National Cancer Institute Preventative Oncology Career Development Award. Dr. Giuliano's career had its inception in the relationship between human papillomavirus infections and cervical cancer in women, and has evolved over the past 30 years to encompass HPV and penile, anal, and oral cancers in men, as well as other infectious diseases and their causal relationships with various cancers. Her work has contributed significantly to our understanding of the rate at which HPV infections are acquired and cleared, the proportion that progress the disease, and also to HPV vaccine protection against multiple diseases in women and men. An expert in the field of cancer research with a long-standing and successful record of National Institutes of Health funding since 1990, she has been actively involved in cancer epidemiology and prevention studies conducted among diverse populations in the United States and abroad. In the course of those endeavors, Dr. Giuliano was a contributor to the 1999 Institute of Medicine reported titled The Unequal Burden of Cancer in the 2005 International Agency for Research on Cancer report that concluded for the first time that HPV is a cause of multiple cancers in women and men. Dr. Giuliano has been honored to receive 2 important awards from the American Cancer Society. In 2013, at the ACS 100th anniversary, she was the recipient of the ACS Distinguished Achievement in Cancer Award. And in 2018, she was elected for the ACS Clinical Research Professor Award for Prevention of Infection-Related Cancers. Dr. Giuliano has authored more than 400 peer-reviewed publications. Welcome, Dr. Giuliano. We're excited to have you today. And with that, we're going to go ahead and turn it over to you and start the presentation.
Okay, Robin, thank you so much for that introduction and welcome everybody. So you're probably all wondering, how does a cancer epidemiologist who focuses on viruses do a talk related to coronavirus? And that is a good question because it's a story of what the past year has been for I think for many of us. Anyway, with respect to my disclosures, I actually have pharmaceutical funding for the coronavirus research, some of which I'm going to be showing today. And as you heard, most of my career the predominant portion of my career is with another virus, which causes cancer, which is a human papillomavirus. And I have several different pharmaceutical relationships around the virus studies, as well as the vaccine that protects against that virus. But today, what we're going to talk about are really 3 objectives. And I'm just going to ask you to bear with me because, like everybody else, we are learning as the virus progresses through the community in the last, it hasn't even been a year for us here in Florida, almost a year. The first is to describe the rate of SARS-CoV-2 infection in the county that our cancer center is located in, which is Hillsborough County, and compare those rates to the state of Florida and also to the United States. And then the second is to compare the prevalence of the antibodies to the virus. So not the virus itself, but the immune response and the proportion of Hillsborough County residents who actually have a circulating antibody based on the study we're doing. And then finally, to understand the landscape of the vaccines that are currently licensed for emergency use and as well remain in phase 3 trials and earlier phase trials to protect against the virus as well as the disease COVID that the virus causes. So just by way of introduction, March hit, March 2020 came and it became really clear that we had this virus that was circulating. And our Cancer Center leadership became very concerned about how the underlying infection in the broader community where our cancer center is located would impact our cancer patients. And in particular, we're really concerned about what % of our patients would become positive. And then, of course, we consider cancer patients to be at very high risk for disease and severe disease following infection. But another part of that question was, what can we expect from the future in terms of how rapidly people are becoming infected, what proportion, and then will we ever be able to achieve what all of you have been hearing in the media, which is herd protection. And again, herd protection really wouldn't happen until around 60 or 70% of the population had circulating antibodies, so protective antibodies against the virus. So we said, look, studying the virus itself can be very difficult in time because, at any one time, people could be exposed, could be infected, could have cleared. And so what we thought the better approach was to really be looking at the antibody response. And I have to say, the reason we took this challenge on is, the Center of Excellence that I lead is focused on viruses and has a long track record of testing for viruses, testing for antibodies. So we were probably the best research group within our cancer center to actually tackle this question. So how did we do that? Where did we start? And I have to say that the data that were reported through the Florida Department of Health and were updated every single day were incredibly important and useful for us as we were designing all of our studies. And I have to say also, as we were monitoring what was happening in our community and in the state, so we understood what precautions we should be taking in the cancer center with our patients and with our faculty and staff. So what I'm showing you are the data coming from the Department of Health. And so let's look at Hillsborough County. Now, remember, the first case of SARS-CoV-2 was, I think, reported on March 2nd. And so this is when we started to really monitor what was happening and the exponential growth in the number of people who tested positive. So I just want to remind you, Hillsborough County has approximately 1.5 million residents. You can see we're located here in Central Florida. And we're really part of a larger community that's called the Tampa Bay community that does include Pinellas and Pasco counties. So as of January 24th, so we had to kind of put a close date to when we were going to report the data for this talk. As of January 24th of this year, 2021, there were over 96,000 known cases of SARS-CoV-2 among our residents. And when I say known cases, that means that there was testing of that individual that led to a positive test result. Now, I want to remind you that when the pandemic started in March of 2020, we actually didn't have routine testing available as widely as we have now. So one of the questions that I think is really important to address is really how many people truly were infected. And because of the way the testing rolled out, this 96,000 is likely an underestimate. And I'm going to show you some data to support that assumption. So what does this mean in terms of the proportion of our county residents who tested positive? Well, if you divide that 96,000 plus by 1.5 million, you get to a prevalence of 6.4%. And when I say prevalence, I'm talking about period prevalence. From the time that the pandemic started until January 24th, counting all of those cases that occurred at each of those different weeks in that time period by the denominator. So you can imagine the percent of new cases per week is actually pretty low. And this is something that I want you to remember also when we talk about the phase 3 trials of the vaccines and the kind of sample sizes they needed to enroll to be able to definitively make a statement about whether the vaccine protected against infection or not. All right, so here are some more data that are reported by the Florida Department of Health. And again, I have to say, these have been so informative. This pattern that I'm showing you has really stayed constant from the very beginning of the reporting. So on the left side, you see all cases. And what you see is all cases by the age group. Now, what's really clear from these data are that most of the cases are occurring in relatively young people, so ages 15 to, let's say, 54, right? And we have, certain peaks. So the new cases are really peaking in individuals ages 25 to 34. But what is very concerning is that although the number of cases, of new cases of virus, of people testing positive for the virus, is actually quite low among older people, the proportion who actually then have severe disease that results in hospitalizations is quite high. And this is what you're seeing here. So a lot of infection in young people, very little hospitalization in the younger people, almost no deaths, but all of the hospitalizations and deaths occurring among older individuals. So the risk of progressing to severe disease, if you are in the age group 55 and older, is very high compared to the younger age group. And what we can see here is that it's really 95% of all the deaths due to COVID occurred among people ages 55 and older. And this, of course, is a really important reason why when we develop public health strategies for who to protect and who to protect first, you can understand the rationale of the governor to start vaccinating at age 65. Rather, the governor did that ahead of the country. When the country had originally said only people 70 and older would receive vaccine, he looked at the data and very wisely said, we need to start at 65. Okay, so how does Hillsborough County compare to what we're seeing in the rest of Florida? Well, as been reported in the media over and over again, the Miami-Dade County area has been a hotspot for the virus and for severe disease, and really remains that way. So that's that dark blue here in the southeast part of Florida. So our area in the middle of Florida is really doing much better. And then if we look at Tallahassee, where you guys are in the Department of Health, are looking even better. And it almost sort of tracks along with population density, which isn't surprising. So if you have very dense populations living in a city, for instance, it's a lot harder to control exposure. As much as, we wear masks and we try to stay 6 feet apart, in some areas of Florida, as well as in the country, it's almost impossible to stay away from people. So now, as far as Florida is concerned, there have been 1.6 million plus cases, test positive cases of the virus, and most of those are in people who are actually Florida residents. So you remember, we have a lot of, snowbirds coming in or people visiting and so on. But the vast majority of those positive cases are in actual Florida residents. And so when we do that simple math again, we have over 21 million people living in Florida. What we have for the state of Florida is a period prevalence since the beginning of the pandemic until the end of January. 7.6% of Florida residents have tested positive. So that's higher than what we see for Hillsborough County. But it's really driven by the Miami-Dade County area, which very likely has double the test positivity rate than what we showed you for Hillsborough County. All right, so when we look again at the breakdown of number of cases, hospitalizations, and deaths by age for the entire state, again, you see a very, very similar pattern where the majority of test positive cases are in the younger populations, ages 15 to 54. And as the populations age, 55 and older, you see the number of new cases significantly drops, but what increases by quite a bit are the proportion of those cases that require hospitalization and those cases that pass away. And again, 95% of all the deaths in the state of Florida due to COVID are in individuals 55 and older. So in the United States, here is a map of the infection prevalence. And again, I just want to remind everybody, in the beginning, really in the first half of 2020 and through the summer, the testing was so variable. People either couldn't access the test or in some areas they were able to access with no problem and were repeatedly tested. And then the policies about who to test and who not to test changed over time. So it's really hard to get a true estimate of the period prevalence, the number and the percent of people in the country who were actually virus positive. So what, and I don't know if you remember in the very beginning, the guidance was only people who presented with symptoms were to be tested. And then we really opened the floodgates and provided testing more broadly to help stop the pandemic. So in the United States, again, as of the end of January, we actually had 25 million test positive cases. So very similar to what I showed you for Hillsborough County and for Florida, around 7.7% of United States residents actually tested virus positive, so 7.7%. So it's not a hugely common virus, and I say that because of course I work with human papillomavirus, so I'm used to hearing percentages like 80% of the U.S. population at one point or another in their lifetime will have been exposed and infected with HPV. However, the death rate due to HPV is minuscule compared to the death rate associated with this virus, and again, particularly among people who are elderly, the most vulnerable. So when we look at the United States, we actually see a slightly different pattern. So we see that the percentage of individuals who are testing positive seems to have two peaks among people who are ages 18 to 29. And this probably represents, you remember in the summer, people were going out in the fall in colleges, university campuses. It was a free-for-all. And then in a slightly older age group, ages 50 to 64, and I wonder if it's this younger population coming home to live with their parents who then became infected. But the death due to COVID, again, looks very similar. The pattern is the highest percentage of deaths are in the age group, really 65 and older, the way these data are broken down by age, where the trend starts to rise at age 50 and really peaks in the oldest age groups. So this is a pattern we're seeing in our county, in our state, and across the country. And again, it's driving our public health policies as to who we most need to protect and protect first. So going back to that story, so March came, COVID started to wreak havoc for all of us. We said, look, what are we going to do? How are we going to understand how widespread the virus is? And I just want you to remember in March, it wasn't clear what percent of the population was infected or would become infected or were protected by natural immunity. And we didn't know how rapidly this would rise. Was it going to be exponential growth? Was it going to be slow growth? So what we said is the best thing we can do is to do a study that really gives you kind of a summary measure. And that summary measure is the antibody. And I want you to think about this. So a person becomes exposed to the virus, they become infected. And if they recover, they develop an antibody. So if I measure the antibodies in a population, that is the summary measure of all past infections. So I don't have to continually test and test and test. What I can do is monitor the antibody prevalence over time throughout the year. And that in one population, and that'll tell us, one, what was the percent of the population that had been infected, had been past tests, not currently infected, and the percent that may have protective immunity. So what we did is in the middle of April, we submitted for funding a grant proposal that essentially asked to do this, which is to study a total of 1,200 Hillsborough County residents randomly selected to ensure that we had 300 in each of 4 age groups. Because we wanted to make sure that, one, we weren't biasing our sample, like, it's just my friends on Facebook or people who go to Publix. And we wanted to make sure that we had people across the age continuum. There are a lot of questions, and there still remain a lot of questions about the ability of an individual who has been exposed and infected to actually mount an immune response and how that varies by age. And of course, we're trying to get balance of males and females. And so then what we're going to do is estimate the percent of residents that have an antibody. So again, not testing for the virus, but testing for the antibody. So one of the benefits of working with HPV is, we're a small community and we all have very similar laboratory methodology in terms of how to measure a virus, but also how to measure antibodies. And as luck would have it, a very close colleague who is at the Frederick Lab at NIH had been charged with developing the antibody assay for SARS-CoV-2 to use as the standard with which to verify and validate all of the antibody tests that were commercially available. So they were sort of the laboratory that helped FDA to make decisions about which tests to essentially issue permission and licensure to go commercially. So we knew we were in good hands. They shared their assay with us as well as some reagents. We quickly set this up in Dr. Sherry Pillen Thomas's laboratory, who's a senior immunologist, and she co-directs the Center of Excellence with me. So we had the epidemiology, we had the laboratory, and then what we needed to do is actually get the population enrolled. And so we're in the middle of the study still. We're trying to quickly complete the 1,200 before everybody starts getting vaccine. And the point was to determine the factors, demographic, exposure history, race, et cetera, that's associated with having an antibody. Among those who test positive, we then moved to what's called a neutralization assay to see whether the antibody actually neutralizes the virus. And then we also follow those individuals at month 1 and month 4 after the first test positive for the antibody to look at duration of naturally occurring antibodies. So where are we? Here's the 1,200 that we propose to enroll. We ask people to complete a brief questionnaire. We collect a serum tube for antibody testing. They test negative, we thank them, we share the results with them. If they test positive, we thank them, share the result, and ask them to come back for 2 additional blood draws, as you can see here. And at those additional blood draws is when we actually collect another tube of blood to look at the B and T cell repertoires to see the specificity of the immune response to this virus. So where are we? Here's, the other thing too, I want you to remember, we're a cancer center, so we're in the business of protecting our cancer patients. So we didn't want potentially virus positive community members to just walk into the cancer center facility where all of our very vulnerable cancer patients are. So we set up a mobile unit with phlebotomists, and this is our bus. It's actually the Mold Patrol bus that we refitted for this study, and the amazing staff that have been operating this study since the beginning. So we actually have 4 phlebotomy stations located in the bus. Everything for registration is online. So we're able to minimize contact with individuals and prevent any kind of infections by accident. So as our enrollment started October 14th, and this is a good story of how long does it take to get a study off the ground. So we submitted the grant proposal April 14th. Okay, so really quickly after the pandemic hit. The grant then had to go under peer review, was accepted, and then it had to go to contracting. It took us that many months to get through the entire process, including IRB approval, and we finally got started on October 14th. Now, we thought when we first proposed this study in April, we thought the pandemic would be over by the summer. To our great surprise, that of course did not happen, and I'll show you some of the data looking at infections over time. As many of you recall, we ended up having our true peak in the summer. I think what we experienced in Florida was just a little warm up in March and April. But the number of cases just skyrocketed in June and July and August, which in a way made the timing of the start of the study a little more fortuitous, because now, again, we were looking at the summary measure of all past infections. So we could really get a much better idea of what had happened through those two big peaks of infection in Florida. So we, as of a couple of weeks ago, we had 634 that enrolled. We'd already completed testing on 466. And I want you to notice the antibody positive prevalence is 11.2%. And you'll notice that that's higher than the 6.7% I showed you for Hillsborough County. And in fact, I didn't update the slides, but as of last week, we're now at 12% antibody positive. Now, this makes sense. Remember I said there were all these problems with getting testing in the beginning of the pandemic. And then there were people who, knew they had COVID. It was obvious from their symptoms and didn't get out of bed to go get tested. So there's always that fraction as well. So we're estimating that around the proportion of our county residents who actually truly were positive for the virus is twice as high as the test positive rates that we've been seeing. So we're at around, again, 12% with the updated data, okay? But still, as you know, that's a far cry away from 60 to 70%, which is what would be needed for herd protection. So let's look at some of the published data on this point. So here are data coming from New York City, and just follow my logic with me. These are the number of confirmed cases at the top, and let's just focus on the cases. So these are individuals who had a nasal swab or, the saliva test and had presence of the virus in that test. They tested positive. So you can see in New York, their big peak was, March, April, and then it came down, things warmed up, and, their infection rate stayed low. Of course, in Florida, we went back up again here, okay? So now let's see how that translates to the antibody. Now, again, the antibody is a measure of past infection. So it makes sense that while the infection was roaring, it probably takes around 2 to 3 weeks to develop an antibody after the infection. And you can kind of see that the shift in time for the antibody to rise is in the latter part of April, the beginning of May. So 2 to 3 weeks after this peak. And because the infection rate dropped, there were very few additional cases that occurred. So the seroprevalence stayed constant, right? We're not adding new cases. And so you can see here, it stayed relatively constant. The little blips are probably just measurement error and, who they happened to sample at that time. Now in New York City, that really experienced a horrible pandemic in the spring of 2020, their seroprevalence is around 20%, 20 to 22%. So it is higher than what we're seeing in Hillsborough County. And I would almost guess that the Miami-Dade County area is probably seeing a seroprevalence very similar to New York City. Because remember, they had the same peak in the spring, but then they had that second peak like the rest of us in Florida did also. But still, a far cry. And remember, it was a horrible, horrible exponential rise in cases in New York. And still, not a high enough prevalence to achieve herd protection, even in the hardest hit area. And this is just showing you for the United States, again, some of the estimates. And I can tell you this is an interesting analysis. They take infection rates and they come up with a formula to estimate what the antibody prevalence would be. So it kind of looks like what we'd expect. The highest prevalence would be in the densest parts of the geography in Florida. Here's LA County. Here's Houston and Dallas, and then again in the Northeast. And this is just showing you, it's another way of looking at infections by state. Of course, Florida kept changing. You know, if you watch CNN, you know, for a while we're doing okay, and then we look horrible and we're the brunt of all the jokes. Georgia became the brunt of the jokes. You know, but the truth is the virus hit every single state at some point. And at some point, one particular state happened to be the worst at that time. So this is, you know, what I like to say similarly with HPV infection, it's an equal opportunity virus. It's going to hit everyone who's exposed, you know, who remains unprotected. The difference is who is going to die from the infection. And that, again, are the elderly people that I showed you and people with severe underlying conditions. So how are we going to get to herd protection? Well, obviously, even after all these peaks of infection, even in the worst hit areas like New York and Miami, we still are not going to get to herd protection. The only way to do that is with vaccine. And, you know, for those of us who work with infectious diseases, you know, we could have told you that. I mean, it's especially when you have such a short period of time and so many people are dying, you really have to get aggressive with measures to achieve that herd protection. So there are a lot of different ways of studying this, and you've probably heard in the media that, you know, the vaccine trials were designed to test for you know, preventing disease or preventing death and so on. So let's kind of step through it. So obviously the thing that most of us care the most about is to prevent infection, right? So I want to know that if I'm vaccinated and I'm exposed to this virus, that I will never become infected, okay? Another way of studying the virus is to say, well, maybe you get infected even though you're vaccinated, but the vaccine prevents the progression of the virus to severe disease. And severe disease would be defined as preventing hospitalization, preventing intensive care unit after hospitalization. And then finally, you know, what we really care about, especially for older populations, is to prevent death. Now, this keeps being discussed over and over again, prevent transmission. So I take a probably a naive view of this, and it really is born from the work that I've been doing with the HPV vaccine and HPV. It follows that if you prevent infection, that you therefore prevent transmission. Now, how does that work? I started by saying that our community survey is not only going to look at the antibody, but it's going to look at whether the antibody neutralizes the virus. So what does that mean? I want to know whether the antibody binds to the virus in such a way that the virus can no longer enter the cell. If the virus cannot enter the cell because it's bound down by antibody, then it cannot prevent it, it cannot cause disease or death. And if it is bound by antibody, the virus bound by antibody, when it's transmitted, it's not possible for that virus that is bound to antibody to enter the cell of the person that I transmitted it to. So it stands to follow that if the vaccine leads to an antibody that neutralizes the virus, then and prevents infection in the vaccinated individual, it should also prevent transmission. Now, the reason people aren't willing to make that statement definitively is that we don't have those data. And actually, we only have indirect data for that phenomena with the HPV vaccine after years of deploying the vaccine in large populations and observing this, which is what actually has been observed in Israel. If I vaccinate 50% of my population, if the vaccine only protects the person vaccinated, then I will see a 50% decline in infection prevalence or incidence. But if the vaccine leads to what's called herd protection is essentially defined as I am preventing transmission to an unvaccinated person, then the percent drop in prevalence or incidence of the infection is greater than the percent vaccinated. And in fact, that was what was just reported for the country of Israel, where they made the statement that vaccination reduced transmission. So it's too early to be able to study that in the United States because we've only vaccinated around 8 to 10% of our total population. But over time, those are the kind of data that we will be able to get, and we will then be able to better understand the true herd effect of the vaccination and then obviously transmission. Okay, so where are we? Well, it turns out, you only hear about Moderna, Pfizer, AstraZeneca, and Johnson & Johnson, but there are actually 40 different products, 40 different vaccines that are in what we call phase 1, safety and testing. Some of them have dropped out. So one of the Merck vaccines has now dropped out. It's not performing the way they had expected. And so they're no longer moving forward with that one. There are 24 in phase 2 and 20 in phase 3. And I'm going to show you those. 8 that are actually in limited or early limited use around the world. And of course, here in the United States, we have our 2, which is Pfizer and Moderna. So here's the picture that I've been looking for. I'm sorry, you had to look at my hands. So here is the virus and here is the antibody. Now I want you to imagine if you have a really high antibody titer, all of these spike proteins are bound out by antibody. Now, to enter the cell, you need the spike. So if the spike is bound out, it cannot enter the cell and it cannot do its nasty deed. All right, so what are the phase 3 trials in limited use? So we have Moderna. It's the new technology, mRNA, that's encapsulated in a nanoparticle. As you know, we've already been disseminating vaccine pretty widely here in Florida. So both Moderna and Pfizer have similar technology. I want you to see the sample sizes. Pfizer enrolled 44,000, and they continue to follow these individuals. And I think Moderna had around 30,000, ended up enrolling in multiple countries. And you can see the efficacy is very high. It's wonderful. Johnson & Johnson is a viral vector. So this is a non-replicating modified adenovirus replicon that is then bound to the spike protein. So this is a protein-based vaccine. And they are also showing very nice results. AstraZeneca, which you've heard quite a bit about, especially now coming from South Africa, they're saying the very similar technology. It's an adenovirus replicon. It apparently is not protecting against the South African variant. I haven't actually seen the data to see how low that protection is. And then we have a protein-based vaccine, Novavax, that is still in studies. Around the world, there are multiple vaccines, and I just want to highlight CanSino, which is a Chinese vaccine, has already been in use in China and has now received approval for use in Brazil. And in fact, my colleagues in Brazil have already lined up and received their first dose of the CanSino vaccine. And you can see there are others as well. This is very good. We need as many efficacious vaccines as we can get rapidly so that we can have enough doses for the world. I just want you to understand that manufacturing vaccines is different than manufacturing drug. It usually can only be manufactured in specialized facilities, and there aren't that many. We don't have as many vaccine manufacturing facilities as we have drug facilities. So the more companies that are in the market and can produce rapidly millions of doses, the more likely we'll be able to get the world vaccinated. Right now, the poorest countries of the world, we don't know when they're going to see vaccine. And although that may not sound horrible to you, you can't go to a world economy until the world is safe from this virus. So although we may achieve herd protection with vaccination in the US, hopefully by the summer or fall, until the world is vaccinated, we're not going to get back to routine business internationally. So trying to get to the next slide. There we go. Okay, so I just wanted to show you some of the data for the Moderna vaccine. This is a 2-dose regimen. This is the vaccine we've been receiving here at Moffitt and vaccinating our patients with. What I'm showing you are not the efficacy data. I'm going to show you the immunogenicity data. I want you to see how strong an antibody response is to this vaccine. And I want to remind you that the 100 microgram group is the dose that was chosen to move forward for licensure. And so they tested 3 doses. In the prevention setting, what we try to do is pick the lowest, most efficacious dose. So remember, we're delivering vaccine to healthy people. So you want to minimize any side effects. And if we look quickly, sorry, there's a little delay. There we go. For those of you who've received the Moderna vaccine, this will come as no surprise. It's a really strong vaccine. So one of the things that we've been noticing here at Moffitt is if you look at dose 1 versus dose 2, that a lot more people have adverse events after the 2nd dose, and they're pretty good adverse events like fever, chills, and some people even reporting pretty moderate experience. Also, headache is more common after the 2nd dose, and a lot more people reporting moderate, not mild, but moderate side effects. And the same thing you can see here, the pain stays the same, but in terms of erythema and redness, local symptoms, nausea. I was in that category, and just plain myalgia. You can see significantly increases with the 2nd dose and becomes more severe. So just to warn everybody, but let me just tell you, it's worth it, especially if you're 50 and older. And so what is that antibody response? So here's day 1, the vaccine is given. 2 weeks after the 1st dose, you already see a really significant rise in the antibody titer. And I just want to show you that we are in a log scale. So it's now titers at 10 to the 5th. So it's, 3 orders of magnitude higher than what we saw prior to vaccination. So 100 and 1,000-fold higher. Now here's dose 2 that's given, and you can see how high after dose 2 the antibody titers go. Okay, so this is a lot of antibody that is circulating to make sure it is binding any virus that your body encounters. And so what they did was they looked at antibodies to the spike protein as well as to the receptor binding domain of the spike protein. Okay, so very similar results. So what about older people? Do older people develop an antibody response that's similar? So here's a classic population enrolled in study, 18 to 55, and you can see antibody titers on the order of 10 to the 6th. More variation in people 56 to 70, but no shortage of antibody production also achieves that high titer, 10 to the 6th. And the same even for people who are 71 and older. And this is really the best news, because remember, this is a group where something like 50% or 70% of all deaths were occurring in this age group. So again, if we can protect with vaccine, it's a real win. Little delay, sorry. Trying to get to that next, there we go. Johnson & Johnson is potentially a one-dose regimen, which I actually think is exciting and, maybe best utilized with younger people. They had some interesting results. Here's a low dose and the high dose group. Again, you'd always want to go with the lowest effective dose. Now, just stay with me. It's a little complicated. So here are people 18 to 55. Here's the first dose before the first dose. Here is 28 days after the first dose, and you can see titers are rising very nicely. A lot of variation, though, probably a lot more variation in those titers than we saw with the Moderna vaccine. Now, here's 28 days after a second dose, and if you compare this second dose titer to the same time point after just one dose, doesn't look different. So this is the suggestion that maybe one dose in this younger age cohort would be sufficient. Now here you see the one dose in the older group, and it just doesn't seem to achieve the same titer. So the number of doses may need to be different for different age groups for the Johnson & Johnson vaccine. And what I'm showing here, I don't want to spend too much time on it, is just the percent of that titer that's neutralizing. And just, glancing at the bars, you see that the pattern looks very similar. So if they develop the antibody, most of it is a neutralizing antibody. So that's the good news. So, work never ends. We have a new CEO, Patrick Wu, who joined us. I have a problem here on the screen. Anyway, so very quickly, he said, look, we have vaccine. It's a new world. We need to vaccinate our patients. We have no idea whether the vaccine is going to prevent infection and result in an antibody titer that's similar to individuals who are not living with cancer. And remember, this is a particularly vulnerable group. So we said, okay, we got enough doses to start vaccinating our patients the first week or second week of January. And we quickly layered on a research study on top of the vaccine dissemination program. And so the aims were to look at the percent who seroconvert after each vaccine dose and by immune status. We have a lot of patients by virtue of their disease are immunocompromised, and by virtue of the therapy they're receiving are immunocompromised. And there are various levels of that immunocompetence. We wanted to know whether that antibody was neutralizing, and we wanted to know what those titers were, very similar to what I showed you for the Moderna and the Johnson & Johnson vaccine trials. What do they achieve after one dose? What do they achieve after two doses? And how does it compare to what was reported in the Moderna trials? And then how long does that antibody last? So here's just a pictorial. We collected blood before their first dose of vaccine. In fact, today is day 28, the first day of our day 28 patients. We're collecting blood before the second dose. And then we're asking patients to come back so we can draw blood again 1 month after receiving the second dose. In April, we will have completed all of the sampling. We run our antibody test, and then we follow patients prospectively. So to date, we've enrolled around, this number is actually a little lower. We actually have 690 patients enrolled, and we did that over a 7-day vaccination period. It's a miracle of miracles when you get an entire research and clinical team all on the same page to get the job done. So we're excited about this. Again, this is the start of day 28, second dose. And here are some of our staff members. We were so busy, we didn't have a chance to actually photograph everybody. So you see some of the clinic staff, the registration staff, the phlebotomists, the people managing the specimens. It really took an army to get this done. And with that, I want to thank you, and thank you for listening, and open it up to questions.
Hi, thank you, Dr. Giuliano. That was a a wonderful presentation that really provided some great information for us all to be able to understand. I'm Bonnie Gaughan-Bailey, and Robin DeWalt, who started us off, I am now taking us to the finish line. I'm Bonnie Gaughan-Bailey, Deputy Director for Public Health Research. And we do have a few questions, and once again, this is an opportunity. If you want to submit a question, please do so at this time. We do have a few, so I'm going to go ahead and get started. Okay, first of all, when should I be tested for antibodies after an infection?
Okay, so that's a really good question. You know, if you want to know what your antibody status is, there is no should be tested for antibodies. There is a should be tested for the virus. But if you want to know your antibody status, I think the best thing to do is to donate blood. All of the blood donation centers, as part of their program, are testing for antibodies. So you're doing two things. One, you're providing an essential service to your community, and two, you get a benefit at no cost to you. You'll know your antibody results.
Thank you. And that kind of falls into the next question. Where can you get an antibody test? So besides the blood bank, are there other places, types of healthcare facilities that provide that service?
You know, that's a really good question. I would call your provider because depending on the health system that your provider is with, they may or may not have the antibody tests up and running at their facility. So for instance, I know that Tampa General Hospital has the capabilities to test for antibodies. I just don't know what other health systems have that. So it's best to call your provider and ask for guidance. Okay,
great. Thank you. Did you look at with the older population, did you look at the differences in hospitalizations and deaths between congregate living facilities and those at home?
That's another good question. You know, the first cases and the deaths were reported, remember, from Washington State, from senior residence living facilities. Here in Florida, we've actually done relatively well with the residence facilities. I don't have the data in front of me. I would need to look at that to give you an exact answer. But the impression I have is that the percent of deaths by age group did not differ that much by whether the person was in an elder care facility or in a home facility.
Okay, thank you. All right, here's another question. Was there a positivity rate that was noted for someone who had other pre-existing conditions such as diabetes?
A great question. And in fact, that is something that we will be looking at. We're still in the middle of the study, so I can't answer that question right now. But in the questionnaire, we very carefully assessed other, so the classic comorbidities of diabetes, heart disease, lung disease, and so on. So we will be able to address this question. I'm hoping we'll have the study, the enrollment completed by the end of March at the worst, and then we'll start analyzing.
Okay. You really did able to catapult your study in a short period of time after all of the required approvals. So congratulations on that.
Thank you, but I have to say it was so frustrating because here's a pandemic rolling out, and you know, and actually it was record time for a study to, you know, to open a study. Usually it takes a lot longer, but it still wasn't fast enough to be able to truly respond to what was happening. And it just shows you that if we can simplify our approval system, we'd be able to get information out there so much faster.
Agreed. Agreed. Okay. Is there any data on long-term health problems following persons who have had COVID and recovered?
We're starting to hear that now. So, you know, again, when when a pandemic first hits, all you have in terms of length of time of follow-up is those few months. So now we're approaching 12 months after the start of the pandemic in areas like New York City. And those individuals are being followed. And I'm starting to hear reports of long-term sequelae in some. And I just want to be careful that people understand it's not everyone. It is. It seems to be some. And similar to the reaction physically to the virus, it seems like it's all over the place in terms of, you know, how quickly someone recovers and they have no symptoms again, and others who seem to have a longer course with the infection and continue to have symptoms afterwards. And a clear pattern has not yet emerged. It will very shortly, but we just don't have enough time and data to be able to address that.
Okay. We have a question. I'm currently in treatment for my second breast cancer, and I had tonsil cancer in 2018 due to HPV. I have been on the fence about getting the vaccine because of the lack of long-term data. My oncologist has recommended it for me. I'm currently on the waiting list. Now, this is asking for your thoughts, and I recognize that you're not their primary care provider, but that also also piggybacks to another question. When is the best time, if you have cancer, when is the best time for you to get your vaccine, and if you're on chemotherapy?
You know, these are great questions. The last study, and I'm sorry I kind of rushed through it because I wanted to leave time for questions. We don't know what the immune response of cancer patients will be to the vaccine. And it's specifically why our new CEO, Dr. Patrick Wu, advocated for two things. One, we know that cancer patients who are currently receiving treatment are very vulnerable to the virus and potentially to severe side effects, severe disease as a consequence of the virus. And because of that, we as an institution have made a decision that with the doses of vaccine we receive, that we want to make sure our patients are vaccinated and they're protected. But while we're doing that, we also want to make sure that we study the immune response. So we know, but you know, the first priority is to protect our patients with vaccine. So our general opinion at the Moffitt Cancer Center is to vaccinate cancer patients. There are, every case is different though. So what I recommend is that you speak with your oncologist and you ask them for who you are as a patient, whether they think that you should receive vaccine and can they get you access to that vaccine.
Okay. Here's another question. Can you speak to how the potential cross-reactivity in serology testing impacts your analysis?
Right, that's a good question from a laboratory person. So the first thing we did, the reason that question comes up is there are 5 endogenous coronaviruses. These are the viruses that cause the common cold. And so they're circulating and you might or might not have antibodies to it. So when the antibody assays were first developed, it was really important to make sure that there was no binding of our antibody to the, how do I say it? So that I wasn't detecting antibodies to one of those 5 coronaviruses, that I was detecting antibodies to this coronavirus, SARS-CoV-2. So there were a couple of things that we did in the laboratory. One is that we started with what we thought were truly naive blood specimens that were collected and banked a year to 2 years prior to the first identification of SARS-CoV-2. And we use that as our negative control. Now, a lot of people will have antibodies to those 5 coronaviruses, so that becomes our starting point. And then we have what are called positive controls that we know people who were infected with this specific virus. And so by using very well collected and pooled negative controls and positive controls, we were able to account for any potential cross-reactivity.
Very good. Thank you. Thank you so much. And I think that we're going to wrap up this presentation. And if you're in Tallahassee, you know that there is an electrical storm that's approaching. And so before we lose connectivity, I'm just going to say thank you again, Dr. Giuliano. It was really wonderful for you to share this information. And then I will just go ahead and finish by saying a couple of quick things. To get your CEs, you need to complete your evaluation and send it in, and that is for nursing. Our next presentation will be on March the 9th at 10 a.m. titled Overcoming Barriers to Improve Outcomes in Uninsured Patients with Diabetes. This is being presented by Dr. Otis Kirksey, who is from Florida A&M University, has been on the Diabetes Advisory Council, and really a leader in looking at health disparities in diabetes and dedicating his life to make those improvements. You can register for this webinar on the Florida Health Grand Rounds website. And with that, I want to thank everyone for their participation. And once again, and special thank you to Dr. Giuliano. Everyone stay healthy, and we'll see you next month. Thank you so very much.